Good evening, everybody. How are you guys doing tonight? Good, awesome. All right. Well, welcome to FAU Harbor Branch Oceanographic Institute and the John and Barbara Ferreira Ocean Science Lecture Series. My name is Sam McGuire, and I'm the Outreach Coordinator filling in for Gabby this week. For nearly 30 years, this series has been at the heart of our community outreach, and it's your involvement that keeps it going. We're glad to have you all with us and hope you enjoy learning about the great work underway here. Before we get started, I want to take a moment to thank everyone who supports FAU Harbor Branch. I'd especially like to recognize John and Barbara Ferreira, along with those who donate to the Ocean Science Lecture Series and make evenings like this possible. Community support impacts so much of what happens here, from the research taking place behind the scenes to the education outreach programs we share within the community. So as you listen this season, you may find a topic or project that sparks your interest. When that happens, there are lots of meaningful ways to get involved, from volunteering your time or sharing what you've learned with others to making a gift in a way that's right for you, including some of the options you see behind me. If you're interested in exploring any of these opportunities, we're always happy to talk with you after the lecture. I also want to thank all of the staff here that help with this series, as well as our speakers. This program wouldn't be possible without them. Next, a few general announcements. As we get started, please take a moment to ensure that your cell phones are silenced. This season, lectures will take place each week through March 25th. Your registration covers access to all of them, so no need to sign up again. And please encourage your family and friends to join us by signing up as well. Each presentation is also available virtually, so hello and welcome to everyone who is tuning into our live stream. And they're recorded, so if you miss one or you want to watch one again, you can do so on our website. For those here in person, after each lecture, we will be hosting a meet and greet reception with our speaker and cookies in the gallery. We'd love for you to stay and encourage you to strike up a conversation with our staff, scientists, and students. This is a great way to learn a little more about FAU Harbor Branch. And be sure to mark your calendar for next week's lecture on March 11th, which is titled Ocean Optics, Shedding Light on the Ocean Around Us by Dr. Wayne Slade. Which brings us to today's presentation by Dr. Corry-Crowe, which is titled Plague Hunters, the Race to Find the Cause of the World's First Pandemic. Greg is a research professor at Harbor Branch Oceanographic Institute at Florida Atlantic University and a National Geographic explorer. He is a behavioral ecologist and population geneticist who studies marine mammals in some of the remotest regions of the world in an effort to advance our knowledge of these species and their environments, and to find solutions to growing conservation and management issues. As well as working in waters closer to home, he works in the Arctic, where he has led a number of expeditions and research projects in partnership with indigenous communities. His interest in the past and ancient DNA has recently led him to explore a new path, attempting to uncover the cause of past human catastrophes like the Justinian plague at the end of the Roman Empire and the quarantine practices in Venice during the Black Death. Please join me in welcoming Greg.
Thank you very much for that introduction, and thank you all for coming out today, and obviously to our sponsors and the staff behind the scenes who make this possible. And it's been a few years since I spoke on this stage, and clearly what I'm going to talk about today is a bit different to what I've spoken about in the past. And so let's get right to it. So, for whom the bell tolls. The first question you're probably asking yourself right now is, why is a marine mammal biologist studying ancient human pandemics? And that's a question I asked myself several times in the middle of this study. But before we launch into this new avenue of research that I've been involved in, I am going to talk about something a little more just to appease the marine mammal gods and those who are interested in marine mammal work, because there was two other competing talks that I was going to give. One was on play behavior in narwhals in the high Arctic, and another one was on mating behavior in beluga whales. And so I'm going to give you a little video on those first. So we can all say this was a truly ocean science lecture. So one of our efforts in recent years has been to try and get to waters that are very clear, fly drones, finally see what a lot of these animals do. And so this is from the high Arctic in Canada, and we're getting unprecedented views of beluga whales in their natural environment. So you have two moms and calves here. And we've been collecting samples and doing genetic studies as well as behavior. This is a very cool study where we're looking at two narwhals, and there's a third cast member here. There's a fish. When we saw this first, we thought they were going to eat the fish. They were hunting the fish. And then we looked more and more, and it seemed like they were playing with the fish. It seems like they were exploring what is this animal. It's a rare species of fish in this environment. And so we've done a lot of work in trying to understand play behavior in large whales. Whales that have time on their hands get to do really cool things. And then back to beluga whales, where there's a large concentration of bulls here, a large concentration of adult males coming together, hanging out together in these massive groups. And so that feeds into a lot of our research on beluga whale mating systems. So that's the end of the marine mammal oceanographic part. Thank you. So back to my original question. Why is a marine mammal biologist studying ancient human pandemics? Well, there are clues in this slide. There are clues in this picture. And this is a beautiful painting of an Aleut hunter who perfected, the Aleuts perfected an incredible ocean whale hunting culture. And so this diminutive craft is trying to hunt probably a right whale or a bowhead whale, many times larger than the hunter, who has crafted a beautiful waterproof kayak, waterproof anoraks, and aconite-tipped harpoons to slow the animal down. And obviously, on the right, we have a woodcut, famous woodcut, of a plague doctor during the Black Death. And so the real question here is, well, what do these two have in common? Well, what they both have in common is these are from the past. These two cultures, these two situations come from a long time in the past. And so if you're a historian, if you're somebody who's interested in trying to reconstruct the past, these two have a lot in common. And one of the areas I'm most interested in is using ancient DNA to reconstruct the past, not just of ancient indigenous cultures and past marine mammal populations, but now obviously I'm getting more and more into our own story and our own lives that we've lived. And so the rest of this talk is going to be about how we use ancient DNA to tackle these very interesting issues of history. And on one general level, for me, I got into this to try and understand the past. We can use that to help us predict the future. And a big issue in areas where I work, but now globally, is how are things like past climate change, past environmental shifts and catastrophes, how have human cultures and animals responded to those? Well, if we could go and collect samples that are preserved in the permafrost, we could extract DNA and we could extract other potential molecules and try and reconstruct what really happened back then. Giving away the story now. We could also apply this to things like conservation and management. Were populations bigger in the past? Did they have more genetic diversity? Have they lost those? Should we be concerned? Are there clues in how some species went extinct from looking at genetic patterns in past populations? Can we learn about disease? Can we learn about population viability and fitness? And so that naturally brought me to the realm of human history, not something I've ever worked on before, but other people are trying to reconstruct our own story, who we are, where we came from, revisit past cultures. And so there's a vast field out there of people doing this that has always interested me a little from the far. And then it's about discovery, right? That's really what drives all of us, Josh, Wayne, so many of us, is the thrill of just trying to figure out what happened or what is happening. And what I would consider ancient DNA research, it's the nearest thing, in my Irish accent, obviously I spell like an Irishman, it's the nearest thing to time travel that I can think of, where you are transported back to another era, and you are working with material where they're going to reveal their secrets to you. You're going to be the first. They're going to tell you their story. And so it's very powerful. And so what we need to do if we're going to do this kind of work is a special facility. Unfortunately, the powers that be here agreed to help us kit out part of our lab as an ancient DNA facility, to where we could, as I say, most of the time, reconstruct past stories of mainly marine questions, marine species. And a lot of that has to do with working on bone or teeth or hard material that keeps the DNA. Is the best place to preserve DNA, unless you're a dinosaur hunter and you need some amber. Bones and teeth are really what you're after, and you're trying to drill some powder and extract the DNA from that. So, and the Arctic is a particularly good place for this. We did a study a number of years ago, not 3,000 years ago, but a number of years ago, about an archaeological site that was over 3,000 years old, and we were able to reconstruct the species that these Aleut hunters were actually harvesting. And we worked so hard in developing the methods that we're starting to get really good DNA, a really good sequence, and so we feel pretty proud that we can step out on the world stage and say, yeah, we really know how to do this here. We're capable of doing this. And so at the beginning, somewhat interestingly, of the COVID-19 pandemic, we get a call. Somebody contacts us, this lady here, her name is Dr. Reese Jiang, and she is a professor at the University of South Florida. And she had pulled together an international team of scientists and archaeologists, and they wanted to collaborate with an ancient DNA lab on human pandemics. So in the middle of a pandemic, here were a group of people trying to study ancient pandemics. And they were particularly interested in reconstructing the causes and consequences of these past calamities that have befallen us, as a way to predict and understand current, the one we were in, and future ones. Could we help? And we thought long and hard, and 5 minutes later, I'm like, yep, we can totally do that. Not knowing anything about humans, anything about human diseases, but we know something about getting DNA out of bones and teeth. So so the story you're going to hear today is the story of what's known as the first global pandemic, the first recorded pandemic in human history. It occurred towards the end of the Byzantine Empire, starting in 541 CE, or Common Era. So 800 years before the better-known Black Death, the storied death that swept across Europe and killed millions of people, there was this very large catastrophe, particularly in the Eastern Roman Empire. Rome had fallen 100 years before, and the main power of Rome was in Constantinople. And here's Justinian here in all his finery. And even Justinian himself got sick and was close to death and recovered. And so this was a very, very major calamity. And it was centered primarily in the Levant, in Egypt, in the Eastern Mediterranean, and included this magnificent city in Jordan, a city I've never been to, maybe some of you have, Jerash, or Jerasha, as it's also known. Still a beautifully preserved city. So just to give you an idea of the scale of some of these pandemics, if we work back in time from the most recent COVID-19 pandemic of a number of years ago, globally about 7 million people died. And various health methods were put into play, various public health restrictions to try and control the spread of that. And as we know, it was caused by the SARS-CoV-2 virus. If you go back 100 years earlier to the Spanish flu, this was a much more devastating pandemic. It was caused by another virus, the influenza virus, obviously, a variant of H1N1. And likewise, people were developing various public health procedures to try and limit the cause of that. And then, of course, we have the Black Death itself, and mainly in the 14th century, but it lingered on into the 17th century. And vast numbers of people died. Originally, during the Black Death itself, nearly 80 million people died. There was the beginnings of sort of public health kind of behaviors, including the famous plague doctors and the masks. We can talk about that later. And they found out that this was caused by a bacterium spread by fleas, Yersinia pestis, the bubonic plague, also the pneumoniac plague. And that species has usurped the word plague. You think of plague, you think of this guy. But as I say, before then, 800 years before then, there was this colossal calamity as well. 25 million people may have died in the first few years. People have assumed it was probably Yersinia pestis. It probably was what became known as the plague. But nobody knows. Nobody knows if that is what caused it. And just to give you some reference as to the population of the world at that stage, so you can see the scale of this issue. There's about 8 billion people in the world right now. Before then, the Spanish flu, 2 billion, 500 million, and maybe as little as 250 million people. So if 15 million die, that's a lot of people, right? A huge proportion. Anyway, so as I say, Jerash was one of the Hellenistic Decapolis, the 10 cities in the ancient Greek world. It's in present-day Jordan. And it was a very wealthy city. It included a beautiful hippodrome here, like in Rome, for chariot races. So it was a very wealthy city. And it was also at the crossroads of a lot of trade from east to west, and that's really important. And then in 659, there was an earthquake that hit the city, and basically the bleachers caved in on the hippodrome. And so many years later, in the 1980s, people were excavating, and they found a major mass grave under the floor of the bleachers. So they knew these people had died, had been interred hastily before then. And they reckoned that this probably coincided with the Justinian plague, this first pandemic. But who were these people, and what actually did they die from? That was the question we set out to tackle. And so we worked with a team of people. It's great when you work with people who have other skills. We worked with archaeologists. We worked with people working on stable isotopes, proteomics, and genomics. And that's what we were really interested in. We needed to get to the DNA. Could we identify the pathogens, the cause? Could we learn something more about these people? And so the question was, Greg, can you get DNA out of these teeth that we're going to give you? And I'm like, I don't know. I hope so. And so we brought them into our lab. Here's one of the teeth we received from USF. We drilled them in our lab here. And good news, we got DNA. We were like, this is fantastic news. We pulled out the DNA. So we extracted DNA from 5 individuals, from different teeth, from 5 individuals. They gave us samples from 5. And so we were super excited about that, as you can tell. But then the real challenge starts. where you have to sort of read the DNA, you have to sequence it, you have to figure out, well, is it any good? I mean, is it just junk? And so I liken sequencing DNA is like deciphering all the words in the book. You have to make sure they're all in the right order. If they're not in the right order, it's word soup. And if you get them in the right order, it's War and Peace, or it all starts to make sense. So we have to decipher the words in the correct order. But if you're sequencing what we know as a microbiome, this is a very popular term these days, if you're trying to look at an entire microorganism flora that an individual has basically retained in its body, if you're looking at all the species of microorganisms, well, that's like deciphering all the words in an entire library, right? So this is what we were trying to do. And so one of the issues with ancient DNA is it's very broken up. It's in tiny little pieces. It's damaged. So we had to kind of patch it together and before we could read it properly, before we could sequence it. And that's something, ironically, not so ironically, is called library preparation. Josh is looking at me. He knows exactly what that's about. And so we needed to prepare each fragment of DNA for sequencing and then stitch the little sequence fragments together if we were to read the entire species or each book. If we were able to read the book, we had to do that. But here's the challenge. Just like ancient manuscripts, ancient scrolls, this is what our DNA sort of looks like. It's broken, patchy little pieces of DNA. And we're trying to put that together and reconstruct an entire manuscript. So I'm kind of setting myself up here, maybe as a hero or as a villain, so that's why I'm making it sound so difficult. And then all you do is you put it on ice, you send it to your buddies at the University of South Florida, and they just run it on the machine and they give you back the data. Piece of cake, right? Piece of cake. And then it happened. Yep, it happens all people maybe once in their life, if they're lucky, never in their life. I get a call from Race in Tampa. Hey, Greg, the samples haven't arrived. And you're like, sorry? Like sure, they've arrived, right? We overnighted it. And I have to say at this point, there even was this little voice in my head as we were getting this sent out going, you know what? Maybe we should drive this over. This is a bit too risky. We've done it 100 times, but we shouldn't do it. And the next few days were pretty depressing. And the samples never would arrive. FedEx had lost them. And there goes our great discovery. And ironically, the current pandemic had conspired to trip us up in the middle of our pursuit of the oldest pandemic. FedEx no longer had to get a signature because of, obviously, our quarantine restrictions. They just had to drop it off. Well, so we cried. Our happy cowboy turned into a really sad cowboy, and we cried and cried into our beer. And then it was like, okay, well, what are we going to do? Well, the story thankfully doesn't end there, because this would be a really short talk, and you'd all be depressed. So maybe we have enough material left. Maybe we still have enough, and you should always extract more and keep some behind. Keep your powder dry, right? And so this time we went back in the lab, and there was still a little bit of powder left for all these individuals. And without telling anyone, I just quietly went away and did the extractions. Then we did the library prep. Then we got in a car and we drove to Tampa. And then we tried to sequence it. And once you do that, you just have to wait. Are we going to get gibberish back? What's going to happen? What's going to come back? And you wait and you wait and you wait and you have your fingers crossed. And then the story starts to finally be told. The secrets start to be shared. The people working on stable isotopes were like, hey, guess what we found? Led by a guy called Robert Tykot. We know these people that you were working on, Greg. They grew up in different locations. They had different diets. Their diets were fed by fresh water, probably from the Sea of Galilee to the River Jordan. The people themselves have lived very different lives. They didn't all just grow up in Jerash, and their parents didn't just grow up there. They came to this city, and they died together, and they were buried together. And so we were like, okay, so this is really interesting. There is a calamity that happened probably in the city, right? Then Gloria comes along with her proteomics, and she said, well, you know, I found some peptides that do occur in various bacteria. You know, it could be maybe Yersinia, but there could be something else. We don't really know. So everybody's going, okay, guys, you're the geneticists. So what did you find? And what we found, the ping happened at the perfect time. All of these individuals had the bubonic plague. We finally could say, yes, these people who died together and were buried in a hastily convened grave, all died, all had the plague. And not only that, they all had the same strain. So when we compared it to all these strains throughout the entire world, they probably contracted this strain together, probably in that city, and then they succumbed to it. And so there was much rejoicing. Secondly, while they all had this strain, it's a particularly virulent strain. So what happens in some of these pathogens, they have certain genes that confer infectiousness, virulence, and so if you don't have those genes, you're benign, no big deal. You've got Y. pestis, it doesn't make a big deal. And so if you look across here, in a lot of prehistorical samples from way back, there's a lot of these virulence factors that are not present. And some of those detections in some of those prehistoric samples are single individuals buried properly in some cultural process, so probably didn't die from the disease. And then if you look at the Black Death, everybody's got every virulent gene you can imagine. The Black Death strain is the worst. But during the first pandemic, here's Jerash. It is the worst of these other individual detections people have made. So clearly this is a bad, bad strain of the disease. And so this was the first proof, right here in River City, that the world's first pandemic was caused by a highly virulent strain of the plague. And just in the last year, several papers have been published on this by our team. And so in this grand scheme, we were able to answer that question. That's what we were able to do with our team. And this is really exciting because now we can put all that history into context. You know, the fall of Rome, the end of the Byzantine Empire, maybe even the expansion of the Muslim conquest into that region. There were climatic events that people believed precipitated the plague. All of these things now can be put into context because we know what exactly the agent was that caused this destruction. Social disruption, things like that. So you're probably asking, what next? Well, now we've just got a taste for this. So now we're getting really into this field. And we're working on a project with Race and her team to look at the Black Death, and particularly in the Venetian Republic during the period of, during medieval times, because the Venetians were the first to develop an isolating mechanism. They dedicated an island called Lazaretto, which means Lazarus, I guess, a place where you had to stay and recover. It was like an early, what's the place in New York that all our Irish, where? Ellis Island. That's where we all came through. And people were held there for 40 days. Quarantina, 40 days. So even though the Venetians didn't know what germ theory was, they thought it was a miasma. You contracted disease through the air. That's why you have these special masks full of lavender and camphor to prevent infection. Well, they were getting bitten by fleas and they were contracting the disease. Even though they didn't know what was causing all this, they were developing these really effective public health systems. But on this island, there are those who died on the island, and they were buried there. So there's 300 years of sick people and people who were victims of various diseases, not just the plague, on this island. And so we're working with USF people in Italy to try and extract the DNA. 1,500 people, I mean, that's a lot of work. And try and understand the causes of disease. And this is particularly, I won't say particularly personal for me, but my hometown in Ireland, Kilkenny, was hit by the Black Death. A lot of people died. And there was a famous friar, Friar John Klyn, who has the best documentation of the Black Death in Ireland. And sadly, he succumbed to it too. And it's worth reading what he says. He says, there was hardly a house in which one only had died. But as a rule, man and wife with their children and all their family went the common way of death. So that notable deeds should not perish with time and be lost from the memory of future generations. I, seeing these many ills, waiting among the dead for death to come, have committed to writing what I have truly heard and examined. I leave parchment for continuing the work, in case anyone should still be alive in the future, and any son of Adam can escape this pestilence and continue this work, the work thus begun. And so the entire Western world was affected by the Black Death, and so I'm hoping that here at Harbour Branch we can make a small contribution to learning about that. And we've got some of the pieces of the puzzle already together. We have a great team. My gang, some of the key people. Andrea Venello is from Venice. He's born and bred Venetian, who has brought this study to us. Robert Tykot works on the stable isotopes. Gloria Fiera works on the proteomics. And Race and Swami do an awful lot of the bioinformatics from the DNA we generate. And we've started a new side to this, which is citizen science. Right here, we have Dr. Pete Gutierrez, who's sitting right over there. He's a very good friend of mine. He's a retired dentist. He plays a mean guitar. And when he saw the clumsy way we were drilling with drills from Home Depot, he was like, just stop. Just, I can't look at it. Just please stop. And so Pete has kind of come to our rescue, and he's improving the way we can just drill the dentin or drill the enamel or drill the cementum or drill the pulp cavity. And so we're super excited about that. And there's just one last little piece of the puzzle, and that is we're desperately searching for funding, as indeed many itinerant scientists are. And so with that, I will leave you, and if you have any questions, I'd be happy to answer them. Thank you very much.
Thank you.
Yes. That's the question. Everybody who comes to my lab asks, am I going to get this? Luckily, certainly with some of these, with the disease, we're mainly focused on no. The answer would be no. They break down in the environment within a matter of hours to days. But we are very carefully looking at, so for example, we want to make sure that if we're looking at things like anthrax or whatever, we have to get all the right approvals. So thankfully, in most of the diseases, no, but there would be some we would be very concerned about, and we have to get all these extra approvals to study those. Yes?
Have you determined at all that most plagues all start with travel?
Travel. So excellent question. Do most plagues start with travel? Yeah, the movement of people is certainly a big element to the spread of disease. But I think there's a lot of stressors that precipitate the opportunity for a microorganism to take hold. So, for example, with the first pandemic, I mentioned the fall of Rome. That was in, I think, about 487. But around the time the pandemic broke out, there was a period where several major volcanic eruptions happened in the world. And so the northern hemisphere, particularly northern Europe, was plunged into what's known as a little ice age. And it's a very interesting time because crops failed, the skies went dark, people were at a low ebb, health-wise. And interestingly, they think it had a lot to do with the spread of Christianity, because people who had traditional beliefs were like, well, that's not working anymore. Nobody loves us. Maybe this God, the single God, loves us. So it triggers the spread of religion, something I'm very interested in, how that plays into the human history. But it probably compromised a lot of people. And that's where disease also sets in. And then you add movement to that, and it spreads. Yeah. You, and then you. So that question was, would AI help speed the process? Absolutely. We work, a lot of us work with teams within FAU who use AI methods to look at things like microRNAs and gene expression. And so, yes, in terms of piecing the puzzle together, AI could be a much more efficient way. So the way the system is currently set up is we screen all the little fragments against a library of known microorganisms. We don't care if if it sorts out they're all bubonic plague. We have no control over that. For that reason, to main objective. Sorry, you had a question. Okay, that's a brilliant question, because, and I'm going to go back and show you an earlier slide of where your question has a lot of relevance. So in our studies, there is a strain in China called the Tianqian strain, and it was on the steppes in the Mongolian plateau around the same time as the documented plague and these other strains that have cropped up in single burials and then our one in a mass burial. And you can see that strain in the tree is probably quite ancestral, which means that's an older strain. So possibly there was a connection to the east earlier on, that these strains and the Silk Road, Jerash is on the Silk Road. And so, yeah, more work being done throughout Asia, I think would be really revealing. So, very, very good question. Yes. So, I would be honest and say I have some ideas, but I am no expert on this field. But what we know from pandemics is they often play themselves out. And for a particular microorganism to constantly remain and be selected for high virulence, well, then it loses its potency and it loses its host. So I don't know the answer to that, but it's a really good question. So hopefully next time around we'll have more on that. So yes, sir.
Did your work give you any clues how to prevent or how to contain the pandemic?
It gave me clues. It gave us clues about more how the epidemiology, how it spread, how it moved through this probably naive population that had very little medical understanding of diseases. And then as we look at them throughout history, I gave you 4 examples. As public health systems start to kick in, you can see how they're starting to control diseases. And I think that's where Venice forms a very interesting crossroads, where this was one of the wealthiest republics in Europe, but it was also one of the busiest in terms of travel. Someone was asking about travel. So, they started to realize that, well, there's people coming from all over the world. Maybe we should have them stop at this island. And so as they start to develop these methods, and as we know during the Spanish flu, I know I'm jumping forward several hundred years, they're like, now that we have an idea of germ theory, you're going to have to wear gloves, you're going to have to wear masks, because contact and inhalation. Flea-borne. Fleas. The fleas.
Okay, so migration in the air.
Yeah. So, and that was actually, it goes back to this gentleman's question, sometimes a microorganism, a pathogen will make a leap, and a mutation in genes of this pathogen, and in the coevolution with host species, meant that it could get transferred, one form could get transferred via fleas, and that's how it kind of took off. So yes. Sorry if I'm missing people's stuff. So by bioagents and things like that. Well, I would agree with you that tinkering with life and tinkering with nature, can have consequences and be regulated. I think very often these ideas start with people saying, well, I want to build a model to try and understand what's going on in the real world and nature, but we know that there are nefarious applications. So that would be a concern. Yeah, certainly. No, they're somewhere on the road. They're on the road to Tampa. Samples. So, yes. Without doubt. And in fact, there's been a lot of really interesting research looking at much older time frames with Yersinia pestis, with the plague, the bubonic plague, and how it infected sheep, and so how the emergence of agriculture, back to you, probably affected those species and allowed for the transfer to humans as zoo. And then in general, yeah, I mean, there's a lot of work here being done on marine mammals, looking at infectious diseases and the impacts of many diseases on the health. And we're actually doing a study where we're collecting blow from dolphins, so as they swim by, try and collect the aerosol, and trying to do the same microbiome work using eDNA to see what they're experiencing on what's infecting them. So, yeah. Okay. Oh, yes, Pete.
Could you talk a little bit about the difficulty of there's and then there's the infectious DNA, and maybe a little bit about the different climates that
Yeah.
samples are coming from.
It's a very good point. I actually mainly talked about the infectious agent, the pathogen. I didn't talk about the host, right, the people. And so one of the things we're trying to do is we're trying to extract DNA from different parts, in this case of the tooth, and Pete's helping us with that. Because, for example, if we can separate out the different fractions of a tooth, it makes our life easier. We can go after, the host versus the pathogen, and there's methods like PCR can help us sort that out as well at the molecular level. And so, I think there's a whole story behind who these people were, and I think the way we do the work in the ancient DNA lab will help us answer both questions. And then in terms of climate, so generally in colder climates, right, there's less infectious disease, there's less pathogens. And a big concern now in the world is as the climate warms, as the world warms, there may be some older pathogens that have been frozen in time in the permafrost that are going to get released, or there's going to be pathogens that live in warmer waters, but as the waters warm and go to the poles, they're going to now start to arrive in sort of pristine habitats with naive populations. And so we're worried that the spread of infectious disease could be a really major problem for a lot of wildlife species and a lot of ecosystems, because you have these naive populations, which is the story, of course, of human history. This is pre-Columbian research, but the next great wave, in fact, the third pandemic when it comes to Yersinia pestis is happening right now in the United States, because it was the move, even though the numbers dying are really small, it was the movement of this particular agent across the Atlantic into a naive native population in the Americas. So, yeah. Yes. I'll get you. Oh yes, it's quite a dynamic field, and there's been a lot of really great research where people are always trying to have a new landmark. Oh, I found it in an older, my samples are older than yours, and this is 5,000. Well, mine is 6,000. Well, mine's 7,000. And so, there's a bit of rivalry there on sort of, where people are detecting, and it's really exciting. And so that tree, all the rest of that tree has been filled by other people. What makes Jerash so unique is that it's this time stamp, it's this mass grave, it's a calamity that was happening at that time. And we now know what that group of people who were hastily buried under the bleachers in a slightly dilapidated hippodrome, what they died from. So it makes it a little unique, but yeah, it's a very exciting field. There's a lot of people working on it. Yeah, you have a question.
Getting back to the start of your talk, the beluga whales, how are they doing?
Oh, hey, I get a beluga question, yes.
Has the change in the environment, warming waters and all of that impacted?
So it's a mixed bag. She was asking how are belugas doing and has the climate really, or the changing environment. So there is a lot of concern that as we're trying to get our arms around particular problems, there's some populations that are quite small, and there's always concerns about small populations. There's some populations that aren't recovering. While we're dealing with that, the rules are being chucked out the window because the whole place is changing. And so we're trying to, in sort of real time, figure out how is this species and other species dealing with this change, and it's rapid change. And all I would say to you is don't give up on any of these animals. They're really tough. Nature's very resilient. I mean, I kind of feel at times humans were put on the planet somehow to stress test every system. And sadly, we end up pushing a lot of systems to where they break and we go, oh my God, this piece of crap, that thing broke. But if we just take our foot off the gas, I think a lot of systems kind of have great resilience in them. And so I tend to be an optimist, but I won't lie to you. There's no room for complacency. So, yeah. Okay. Thank you.
Well, let's give
one more here.
one
more.
One more.
How does a systemic infection end up in the tooth?
Pete, can you answer that? How does a systemic infection end up in the tooth? It's part of the bloodstream, and it goes through the bloodstream, and it goes up through the pulp cavity, and it takes root in the root. And so we can come along 100, 1,000 years later and hopefully recover sort of all the pathogens along with all the healthy flora. You know, there's a lot we could learn. Some of these people maybe had a generally very healthy life, but it's the bloodstream.
But in those days, how many teeth did the average individual have?
Sadly, I will say some of the teeth we've looked at they had really good dental hygiene, some of them. So they didn't drink a lot of soda. So or highly processed foods, yeah.
All right, let's give Greg a round of applause.
Thank you. Very much.
If anyone has any other questions for Greg, we'll be out in the gallery. There's cookies, and everyone, please join us next week for Dr. Wayne Slade's presentation as he sneaks out of the auditorium.